on developing novel drugs for immunoinflammatory diseases. Neil, the floor is yours.
Thank you, and thank you for having us. I'm Dr. Neil Walker, and I'm the CEO of ACRS, and I'll be walking through an overview of the company for you today. So ACRS is a development stage biotechnology company focused on the kinome and the development of small molecule therapeutics for immunoinflammatory diseases. We have three key components to the company. The people, we have world-class ex-Pfizer kinase leadership with decades of experience looking at the kinome. We also have the Connect platform, which is a proprietary discovery engine that's not only versatile, but flexible enough to look at both reversible and irreversible inhibitors. And we also finally have an innovative pipeline with 3 clinical stage assets, which I'll go through in more detail. So how big is the kinase opportunity? If we look on the left-hand part of the slide, we have over 70 marketed drugs targeting various kinases, driving almost $50 billion in annual sales. The good news is that on the right-hand part of the slide, over 90% of the human kinome remains undrugged, and that's the opportunity that ACRS is going after. To do this, we need strong leadership, and we have a leadership team that has a proven track record in immunology and inflammation, decades of experience at large pharma companies like Pfizer. And in fact, a number of our senior leaders were co-inventors of tofacitinib, which really gave rise to the oral immunoinflammatory space a number of years ago. Also, as I mentioned before, we have a proprietary drug discovery platform called the Connect platform. And what this is composed of is a proprietary chemical library with custom kinase and immune assays, but also importantly, a multidisciplinary team that helps us to decrease the time to lead optimization by half or more. It also allows a high degree of flexibility. We have a very wide array of design approaches specific to each kinase that we're trying to target. So these are a couple of the examples of our clinical stage assets where we've used a few different techniques in order to arrive at the actual drug. Our MK2 inhibitor, or dunsametinib, used in that process, we used a unique substrate selective drug design to come up with that molecule. In the middle column, we used a tissue restricted approach in order to enhance the safety of the compounds, and notably ATI 1777, we developed a skin specific, what we call a soft topical JAK1/3. We also have in the preclinical stage an oral gut bias reversible inhibitor targeting IBD. And then finally, not only do we work on reversible compounds, we also have the ability to develop irreversible compounds. And then ATI 2138 is our covalently bound oral small molecule binding to ITK, TXK, and JAK3. And here's an overview of our current drug development pipeline. As I mentioned, we have 3 clinical stage assets. Dunsametinib is the lead. ATI 1777 is our topical soft JAK1/3 inhibitor. ATI 2138 is our oral covalently bound small molecule inhibitor of ITK, TXK, and JAK3. We also have an earlier program that is we refer to as our gut bias program. It's a JAK inhibitor program looking at inflammatory bowel disease. That's in the preclinical stage. And then finally, ATI 2231 is a second generation MK2 inhibitor that we're looking at preclinically for various oncology indications. So first, a little bit about the lead molecules, dunsametinib, or ATI 450. This is an oral small molecule that's designed to block targets of broadly used biologics. It blocks TNF-alpha, IL-1 alpha beta, IL-6, also IL-17. And by inhibiting these multiple cytokines, we believe mechanistically that dunsametinib has the potential to treat a multitude of diseases that are driven through these mechanisms. And as I mentioned before, we enabled a unique way to go after this particular target. This was actually quite a difficult target to drug. And what we have here is a slide depicting how we capture MK2 in an inactive state. In the nucleus, inactive MK2 and P38 dock in a high affinity complex that exhibits a binding pocket, as we see here on the left, formed by juxtaposed walls of both proteins. And then, as we see on the right, what ATI 450 or dunsametinib does is bind to both walls of the pocket, stabilizes the complex, and thereby prevents MK2 activation. And this is a rather unique MOA that enabled us to drug a target that for many years was extremely difficult to drug. So then we want to understand the selectivity profile. As it turns out, dunsametinib is highly selective. We tested at 5 micromolar versus 193 kinase, which is quite a high concentration, and we demonstrated greater than 350-fold binding selectivity on all kinases in this panel, with the exception of P38 alpha and beta. We also showed that it is highly selective for the P38 alpha MK2 complex versus other P38 substrates, as we see in the right upper panel. And on the right lower panel, we also demonstrate that dunsametinib binds to P38 MK2 complex with higher affinity than either P38 or MK2 alone. We also want to understand its efficacy and safety in a variety of tried and true animal models to support our development in the clinic in various immunoinflammatory diseases. So we conducted a variety of studies in both the well-trodden mouse collagen-induced arthritis model, rat streptococcal cell wall arthritis model, the adoptive transfer mouse model of colitis, and also a murine no-mid model. And in all cases showed a robust effect in inhibiting cytokines such as TNF and IL-1 to quite a degree in all these animal models, supporting the progression into the clinic. So next was doing the first phase of work, which was the SAD-MAD work, where we wanted to demonstrate both safety and also the pharmacodynamic profile that would support the mechanistic basis of going into diseases like rheumatoid arthritis. And then we conducted a phase 2A, a small study, proof of concept over 12 weeks in moderate to severe rheumatoid arthritis. And once demonstrating that proof of concept, in our minds, that would enable us to go after a variety of diseases that were driven through a similar mechanism. So first, a little bit about the phase 1 study with ATI 450 or zunsemetinib. We demonstrated that it was well tolerated through the SAD/MAD work. Linear PK after multiple dosing, and marked cytokine suppression as we had seen in the preclinical studies. We then embarked on a phase 1 MAD extension study as we had no dose-limiting toxicities up through 50 milligrams BID, and we looked at 80 and 120 milligrams BID, and again, showed that it was generally well tolerated. All AEs were mild, and we did see additional cytokine suppression, although it was incremental at these higher doses. And here we see the PK profile, half-life is about 9 to 14 hours. 80 milligram cohort was dose proportional with the previous cohorts. We didn't, however, see any significant increased exposure in this study in the 120 milligram cohort. Here, what we looked at was ex vivo LPS stimulated cytokine suppression, and you can see in each of these panels on this slide and the subsequent slides that as measured at day 1 through day 7 and at day 7 at 2 time points, both at peak and trough, that we showed quite potent suppression. HSP27 is a surrogate for MK2 suppression, and you can see we showed a nice dose response up through 120 milligrams. And you'll see a similar pattern here with TNF alpha on the bottom of this slide, IL-1 beta at the top of this slide, and IL-8 at the bottom. And then, of course, IL-6 here with the ex vivo LPS stimulated model. Same pattern, although a little bit less impressive versus the TNF and IL-1. We attribute that to LPS stimulated model not being the best system to measure IL-6, and I'll show you why in a subsequent slide. Here, what we're looking at is ex vivo IL-1 beta stimulation, so we're using a different model than LPS. And what we can see here, if we focus in on the middle part of the slide, is that IL-6 is suppressed on par with TNF alpha. So as it turns out that the IL-6 suppression is a little bit sensitive to the model that one uses, and we demonstrated that in this model versus the LPS stimulated model. And so at the end of the day, we've definitively shown that zunsemetinib inhibits pro-inflammatory cytokines of interest such as TNF alpha, IL-6, IL-8. And then next, we moved on to our proof of concept study in RA. This trial design was placebo-controlled. It was a 3-to-1 randomization. We wanted to get in patients that were rather tough to treat. We had a screening criteria of CRP greater than 5 milligrams per liter, which is a little higher than the normal range. And we also wanted to make sure that these patients had rheumatoid arthritis, so we used a screen-in wrist MRI to assess the joint for disease. The primary objective of the study was safety, of course. We're also interested in showing the lack of tachyphylaxis, which had plagued the P38 inhibitors in the past. And then, of course, we looked at a variety of efficacy parameters as secondary objectives. So here we see some of the clinical data. This is the DAS28-CRP, mean change from baseline. You can see in the red line, the drug group, we did quite well, continue to decrease over the 12 weeks of the study, getting down to a change of 2. And you can see the placebo group with 2 patients behaved quite nicely as a placebo should, with no real response. And this is the same pattern you'll see in the next few slides. Swollen joint count, median % change from baseline. Placebo group really didn't do anything. And we can see the drug group continue to improve throughout the study down to week 12. Same with the tender joint count, median % change from baseline, same pattern. And then this is the ACR 20, 50, and 70 analysis. So if we look at day 84 in the study through 12 weeks, the ACR 20, 50, and 70 scores were 60% in ACR 20, 33% in ACR 50, and 20% in ACR 70. So this is right where you want to be for a drug at this stage, which is right in the mix from a competitive landscape perspective. And this is finally the DAS28-CRP. This is responder analysis over time. Essentially, if you have a DAS28 less than 2.6, you're in remission. If you have a DAS28 less than 3.2, you have low disease activity. And you can see by end of study at week 12, day 84, we have 40% that meet the criteria of having low disease activity or remission of DAS28 less than 3.2, and then 20% that met the criteria for DAS28 less than 2.6 or being in remission. So quite a reinforcing and robust set of data in this small study. We also wanted to characterize some of the pharmacodynamic effects to buttress some of the clinical data. And what we see here is we wanted to show that there was durability in terms of the cytokine suppression, thereby no tachyphylaxis, which we had already demonstrated with the clinical data. And as you can see here, when looking at an ex vivo LPS stimulated model, looking at day 1 versus day 84, we see the same level of TNF alpha suppression and the same level of IL-1 beta suppression in the beginning of the study as at the end of the study. So that was an important additional piece of information to incorporate into the full data set. We also looked at endogenous plasma cytokine levels, TNF alpha, IL-6, IL-8, and MIP-1 beta. And in all these cases, we showed, again, sustained and potent suppression across the board. Left upper panel, TNF alpha, IL-6, and then on the bottom of the slide, IL-8, which bounced around a little bit, and then MIP-1 beta, which is a marker that's often studied in RA, showing, again, nice sustained suppression from beginning of study to end. From an AE perspective, drug was quite well tolerated, mainly mild to moderate AEs in the drug group, nothing else of note. And then just looking at an overall summary, what we demonstrated in this early small study in RA with 50 milligrams BID was rapid reduction in the median percentage of tenor and swollen joints. Importantly, also showed reduction in DAS28 CRP, showed rather compelling numbers on ACR 20, 50, and 70, and importantly showed CRP reduction sustained over 12 weeks over the course of the study. And again, zunsemetinib was generally well tolerated. We also, at that point, had demonstrated some positive phase 1 studies where we showed no dose-limiting toxicity, marked inflammatory cytokine suppression, and PK data package that supports potential for dosing flexibility. We're currently looking at BID, but also believe we can dose QD in the future. So the next step for that program is a phase 2B program, which is currently underway. Next asset in the clinic is ATI 1777. This is our topical soft JAK. Our goal here was to design a tissue-specific JAK inhibitor so that we could maintain the efficacy and minimize the potential for any systemic toxicities. We also importantly selected the molecule to be JAK1/3 specific and minimize any JAK2-mediated hematopoietic effects. And also, we decided to go after, which is a little non-traditional, the moderate to severe atopic dermatitis population rather than mild to moderate, just given the competitive set that exists in mild to moderate and the market need for more treatments for moderate to severe patients. So this is some of the early data from the phase 2A study that reported out last year. We can show a nice percent change in EASI score, modified EASI score through the 4 weeks of study. And this is from day 0 to day 28. So over 4 weeks, nice spread versus vehicle. And then looking at some secondary endpoints, nice effects on the modified EASI 50, over 90% in the drug group, EASI 75, over 60%, and EASI 90, over 30%. And then here, this slide depicts the soft aspect or the tissue-specific aspect of our drug. This is looking at plasma levels at 1777, basically plotting over 140 data points from all the subjects in the study. And we definitively show that this very little systemic absorption, if any. The average concentration of subjects receiving 1777 was never greater than 5% of the IC50, which you can see at the top of the slide here. So we feel we went a long way to proving out the soft concept of this compound. And again, pretty well tolerated on the AE front. We showed a good safety profile here thus far. One treatment-related AE in each of the ATI 1777 and vehicle arms, which was application site pruritus in the 1777 arm and irritation in the vehicle arm. So what we have going into the phase 2B study is we have a positive proof of concept first in human study in moderate to severe atopic dermatitis with our topical soft JAK. We have a PK profile which supports the tissue-specific approach and rather rapid and continuing improvement over the 4 weeks of the study. Next clinical stage asset is ATI 2138. This is our oral small molecule ITK JAK3 inhibitor. And the way we think about this is it's very similar to cyclosporin, but it's more specific to the immune cells. And cyclosporin tends to inhibit calcineurin, which is more widely expressed, potentially creating off-target effects. So going after the unique kinases expressed only in immune cells allows us to be a little bit more selective. And we are positioning this to go after T-cell mediated autoimmune diseases. This is a rather potent covalent inhibitor. On the left-hand side of the slide, you can see that this is the co-crystal structure of 2138, showing the covalent binding to ITK. And on the right-hand side, you see a little bit of the selectivity and how potent we are on inhibiting ITK, TXK, and JAK3 in cells and also in whole blood. We also conducted a couple of preclinical studies looking at showing the safety and efficacy of the compound in a mouse model. And we showed that ATI 2138 is efficacious in a collagen-induced arthritis model. And you can see on this left-hand bottom slide here that ATI 2138 was more potent than Enbrel in terms of improving the clinical arthritis score. And similarly, when you look at the joint histology score, also performed quite well there, particularly relative to the positive control of Enbrel. And this is really the gold standard model in terms of demonstrating some predictability as you head into the clinic. And finally, we also studied 2138 in a T-cell transfer colitis model. And what we showed here is that ATI 2138 reversed the weight loss trend in this disease model to match naive controls. We also showed that it was effective in preventing tissue damage in the ileum by histology, as we see on the right-hand part of this slide. So what we have developed here at AclarisTx over the last few years is an experienced team with a proven track record of R&D and business development experience at numerous large and small companies alike. We also have a technology platform, a drug discovery platform that's composed of a proprietary drug library in addition to a multidisciplinary team that can quickly target the kinases of interest. And then, of course, we have a developing pipeline with 3 clinical stage assets, a variety of assets at the preclinical stage, and then we're repopulating the top of the funnel with additional discovery assets that are in flight as we speak. So this year, 2022 is a relative execution year. We're gearing up to report out a number of studies in 2023. We have 3 ongoing phase 2 studies with zunsemetinib, the lead. That is in rheumatoid arthritis, which is a phase 2b dose range study. We're looking at 50 milligrams BID and 20 milligrams BID versus placebo. And then we have 2 proof of concept studies in hidradenitis and psoriatic arthritis, where we're looking at 50 milligrams BID versus placebo in those 2 studies. And all 3 will read out in 23. And then we are also moving forward with a phase 2b study with our topical soft JAK, ATI-1777. That will also read out in 2023. That is in patients with moderate to severe atopic dermatitis. And right now we're winding up the SAD work with our covalently bound ITK oral small molecule ATI-2138. We will be looking to report that data out towards the back part of this year, and then head into a MAD study before choosing an indication to move forward with in our first POC study. And finally, we'll be submitting an IND at the tail end of this year with 2231, which is an MK2 inhibitor we designed for oncology indications, and that is moving forward on track. Thank you for your time today, and look forward to reporting out on more catalysts and as time moves on.
I want to thank all our presenters for taking part in what has been a very productive and informative series of presentations. We appreciate the time and effort that went into preparing them. Thank you again from the H.C. Wainwright team. Thanks, Neil.
Thank you.